In this podcast, we will focus on white blood cells that we call phagocytes and their role in removing and killing pathogens. A few of them have an additional function that we call antigen presentation. We'll start our discussion on antigen presentation in this podcast, but we'll continue it in later podcasts. We'll start with some definitions and show how phagocytosis is a particular type of endocytosis. We'll look at the characteristics of professional phagocytes and a couple others. We'll then look more closely at the process of phagocytosis and how opsonization helps this process. Dendritic cells and macrophages are antigen-presenting cells, so we'll look briefly at the process of antigen presentation and how these cells bridge the early and late immune responses. Endocytosis is the process by which cells take in or internalize substances from their external environment. These substances may be solid or liquid. When we speak of cells internalizing particles, such as bacteria or other types of microbes, we use the term phagocytosis. So phagocytosis is the endocytosis of particulate matter. Endocytosis of fluid is called pinocytosis, also known as cell drinking. The endocytosis of particles can occur following specific binding of the particles with receptors on the surface of phagocytes, and this is called receptor-mediated endocytosis. This is what happens when microbes are coated with opsonins, such as the complement component C3b. The opsonins bind with receptors that are present on the surface of the phagocytes, stimulate the phagocytes to enhance their ingestion of the particulates and killing of the microbes that they have ingested. Phagocytes belong to the group of blood cells we call white blood cells or leukocytes. These cells develop in the bone marrow, and there are two main lines of development of white blood cells. One line of development leads to lymphocytes, and another, called the myeloid cell line, leads to the development of different types of phagocytic cells. Most lymphocytes work in the late immune response, except the natural killer cell, which we saw works early to protect us against viruses. In contrast, the myeloid cells start working early and are important in the early immune response. They continue to work with the lymphocytes when these become active. I just want to point out as well that one type of myeloid cell, which is circulating, which is the monocyte, enters tissues and becomes further changed or differentiated into macrophages or dendritic cells. The professional phagocytes include neutrophils, monocytes in the blood, and macrophages and dendritic cells in the tissues, which, as I just said, are derived from monocytes. These cells are very efficient at phagocytosing microbes. So let's learn some other interesting facts about them. Neutrophils are the most abundant leukocyte in the blood, making up about 60% of the total white blood cell count. They are granular cells with multilobe nuclei, usually about 3 to 5 lobes. They are the first white blood cell to leave the blood and enter infected tissues, so that in acute inflammation, and inflammation is the body's response to any kind of injury, in that acute response to injury, we find many neutrophils in the area of damage, of whether it's been infectious damage or toxic damage. Neutrophils have a short lifespan and usually die within a few days of being produced. Monocytes are often referred to as agranular, but they really have very fine granules in their cytoplasm. Monocytes are also impressive phagocytes and are capable of ingesting a range of organisms, but they are also capable of changing into long-lived resident immune cells, such as macrophages and dendritic cells. Macrophages in different tissues have different names, such as splenic macrophages in the spleen, Kupffer cells in the liver, and alveolar macrophages in the alveoli of the lungs, just to name a few. A couple other leukocytes in the blood have phagocytic capacity, but are not quite as efficient as neutrophils and monocytes. And these are the eosinophils and the basophils. Eosinophils have coarse red granules that stain with a dye called eosin, hence its name. These granules contain proteins, damaging proteins, which can help to damage large organisms such as worms and help us to protect against this type of pathogen. Eosinophils are also implicated in the development of allergies. Basophils are the least abundant of the white blood cells in the circulation and appear to have dark blue cytoplasmic granules because they stain with another type of stain called hematoxylin. Basophils can ingest and kill microbes, and their granules contain heparin, which helps to keep blood from clotting. So what are the steps in phagocytosis? First of all, the phagocyte has to find the microbe. How does this occur? This occurs because substances are released from the microbes themselves, released from the activation of complement in the vicinity of the microbes, and also substances are released from damaged cells, cells that are damaged by infection. And all of these substances that are released from this area of infection are perceived by the phagocyte, and the phagocyte moves with a very directed movement, which we call chemotaxis, towards the bacteria. So what happens when it reaches the bacterium? When it reaches the bacterium, it detects it either by the presence of bacterial specific molecules on its surface or by the presence of opsonins coating the bacterium. This leads to the attachment and zippering of the phagocyte pseudopodia, which are like long arms that come out around the bacterium. The zippering of the pseudopodia leads to the bacterium becoming engulfed in a vesicle, which is called a phagosome. Lysosomes in the cytoplasm merge with the phagosome to form a phagolysosome. And lytic enzymes, which are within the lysosome, are in fact poured onto the microbe and destroy and digest it. In non-antigen presenting cells, these digested fragments are extruded from the cells as soluble debris. But in antigen presenting cells, bits of protein from the bacterium will be expressed on the surface of the antigen presenting cell, but more on this in a very short while. Opsonization makes the interaction of the bacterium with the phagocyte an example of receptor-mediated endocytosis. Note that this bacterium in red is coated, and it will be the entire thing that will be coated with C3b, which is the complement-derived opsonin. Note that there are receptors for the complement of opsonin C3b on the surface of the phagocyte. These are shown here as complement receptor type 1 or CR1. The opsonin binds to the receptors, and this facilitates the engulfing of the bacterium by the phagocyte within the membrane-bound vesicle we call the phagosome. As we saw, the lysosomes will fuse with the phagosome, forming the phagolysosome, which will result in enzymic killing and destruction of the bacterium. Dendritic cells and macrophages can also act as antigen-presenting cells. These cells take in the bacterium or the other type of pathogen and break it up into take the proteins from these pathogens and break it up into smaller proteins or smaller peptides, which are smaller strands of amino acids. And these will be used to show to T cells, and that interaction is what starts the late immune response. The dendritic cells has multiple arms or dendrites. The dendritic cells, as we see, takes in the pathogen and degrades it, as I said, into smaller bits of proteins. These snippets of proteins, which we call peptides, are added to these molecules, which are called MHC molecules. MHC stands for major histocompatibility complex. So these small bits of proteins or peptides are put into clefts which are present at the top of these MHC molecules. And the combination of the peptide plus MHC molecules are expressed on the surface of the phagocytic cell, of the dendritic cell in this case. And this combination, T cells this combination of MHC and peptide are shown to many T cells. And those that have receptors on their surface that best recognize the combination of the MHC with the peptide will be stimulated to respond to this particular peptide, which belongs to the pathogen. So we are beginning to stimulate T cells that can recognize the pathogen. Dendritic cells are interesting cells, and they take these bits of protein from the infected tissue. What we're seeing here is skin that has a cut, and we're getting germs coming into the layers of the skin, and the macrophages are trying to fight them off. But in addition, the dendritic cells have taken in some of the pathogens, have started to degrade them, and the dendritic cell is moving in lymphatic vessels to regional lymph nodes. And it goes there to show the bits of pathogen, the peptides, which are going to be shown on its surface with MHC molecules to lots of T cells that are coming through. T cells pass through these lymph nodes. So it's the best way of trying to find the T cell that can best respond to those different proteins from the pathogen. And in this way, the dendritic cell acts as a bridge between the early immune response and the later immune response. This table summarizes the information discussed in this podcast. Stop the podcast and spend some time reviewing this information before attempting the questions provided. Thank you for your attention.